Hello everyone, my name is Louie, and today I'll be talking about my capstone proposal, the conformational change in the SARS-CoV-2 viral entry pathway induced by Kobophenol A structural analogues. Before I begin, I'd like to thank Dr. Goez, my capstone advisor, as well as the chemistry department at Pacific University. Today I'll be discussing four main points. Why is SARS-CoV-2 virus important? How does the virus infect the human body? How does the compound Cobophenol A stop SARS-CoV-2? And my proposal for identifying other inhibitors of the virus's pathway of entry using Cryo-EM. And so why is the SARS-CoV-2 virus important? Well, since December 2019, the human population has been affected by this pandemic, and global infection cases have totaled up to more than 140 million cases, with deaths totaling up to more than 3 million worldwide. Furthermore, there has also been news of 5 new variants of SARS-CoV-2, as seen here. And while there has been rapid development of already 3 vaccines, there has only been one FDA-approved antiviral medication, the RNA-dependent RNA polymerase remdesivir. And so as a result, society is in need of identifying more antiviral treatments for COVID-19 patients. Before we go into the mechanism of infection of SARS-CoV-2, we need to identify the proteins involved in the viral entry. In this case, the S protein of the SARS-CoV-2 virus and the ACE2 receptor of the human host. Researchers have already conducted studies to identify the protein structure of both proteins using Cryo-EM. Cryo-EM, with cryo meaning low temperature and EM meaning electron microscopy, is an electron microscopy technique used to identify protein and molecular structures with resolutions of 3 to 5 angstroms. The first protein of interest is the structural spike or S glycoprotein, which splits into 2 main subunits, an S1 and S2. The S1 subunit is a receptor binding domain, which directly interacts with the host's ACE2 receptor. Meanwhile, the S2 subunit is composed of 5 different components, all which are used to facilitate fusion of the viral and cellular membranes together. The second protein of interest is the angiotensin converting enzyme 2, or ACE2 receptor. And the ACE2's domain of interest is the peptidase domain, which interacts with the S1 receptor binding domain, allowing for entry of the virion into the cell. And so, on this slide is 3 protein structures of the SARS-CoV-2 S protein that are all identified using cryo-electron microscopy. You can observe the S1 and S2 subunits as shown here, as well as the closed, open, and open bound to the ACE2 receptor conformations. And in these 3 different conformations, the protein structure of the S protein goes through a subtle conformational change, but a conformational change nonetheless. For the ACE2 receptor, the Cryo-EM structure is also identified. The left illustrates a refined Cryo-EM structure, and the right is a cartoon representation, more clearly showing the important peptidase domain of the ACE2 receptor right here. And so here you can more clearly see that with the binding of the S1 subunit receptor binding domain of the ACE2 receptor's peptidase domain here, a conformational change is observed in the ACE2 receptor. These Cryo-EM structures ultimately are very important for understanding the conformational change that occurs in order for the virus to enter the host. And so now I will talk about the SARS-CoV-2 mechanism of infection. It involves the interaction between the spike protein and the ACE2 receptor, first beginning with the S protein's S1 subunit binding to the ACE2 receptor of the human host. Next, the S2 subunit activates the S protein, inducing a conformational change, and then the remaining components of the S2 subunit help to facilitate membrane fusion of the virus. And finally, endocytosis of the SARS-CoV-2 virus occurs, as shown here, where the viral membrane fuses with the cellular membrane and enters the cell. And so this leads me to the compound of interest, Kobophenol A, and its inhibitory effects that it has on the viral entry of the virus, as shown in literature. Cobophenol A has been found to block the interaction between the ACE2 receptor and the S1 receptor binding domain in silico and in vitro. So it can inhibit this interaction in vitro with inhibitory concentrations of 1.81 micromolar, as well as it's been found to inhibit SARS-CoV-2 viral infection of cells with an effective concentration of 71.6. The structures here are also predicted conformational changes that Cobophenol A causes on the green S1 receptor binding domain, as well as the yellow ACE2 receptor. And so the takeaway ultimately is that the Cobophenol A compound blocks the S1 receptor binding domain interaction with the host's ACE2 receptor. And so now I will explain my proposal for identifying the conformational changes observed from Cobophenol A analogues. I will discuss the structures of interest, what technique Cryo-EM is, and my proposed procedure. And so these are the structures of interest, as they are structural analogues of Cobophenol A. As you can see, Vaticanol C and Vitazin A all possess aromatic groups that are very similar to Cobophenol A. And so now, what is cryoelectron microscopy? Well, cryoelectron microscopy is an imaging technique of electron microscopy used to look at protein or molecular structures. This is done typically in extreme conditions, typically in a high vacuum environment, as well as very low temperatures of around minus 90 degrees Celsius. Cryo-EM samples must be prepared by rapid freezing of the sample by placing the protein in a vitreous layer of ice. This glass-like ice results when ice is not given time to crystallize, and as a result, it causes the ice to sort of form a glass-like transparency. Afterwards, the sample is processed via a transmission electron microscope, where the electron beam shoots the sample and a high resolution image is collected as a result. And so my proposed method for identifying inhibitory compounds goes as this. The synthesis, first I will begin by synthesizing the S protein's S1 receptor binding domain and the ACE2 receptor individually. From there, I will isolate these proteins using different techniques such as size exclusion chromatography and ultracentrifugation. Then I will combine these proteins together along with the structural analog of interest, either Vaticanol C or Vitazin A, and allow them to incubate and form the conformation, hopefully, that will show the inhibitory effects of this structural analog. From there, I will create the Cryo-EM sample as shown here in this infographic. I will apply the sample, blot the sample, and then rapidly freeze the sample using liquid ethane. Next, I will shoot the sample using a transmission electron microscope and then optimize for conditions that will allow for high resolution images. Data collection will then ensue, and I will process these 2D images into 3D images using CryoSpark, a software program. Finally, I will try to observe a conformational change and see if this structure of the structural analog, when it binds to the ACE2 receptor and the S1 receptor binding domain, if it varies compared to the typical conformational change that arises when the S1 receptor binding domain binds to the ACE2 receptor. A difference in conformational change means there is potential for an inhibitor in vitro. And so this is what a proposed result, what the results could look like. I would have high resolution images as shown here, and I would superimpose them together to create a 3D structure in the hopes that I can sort of identify the conformational change that occurs. as a result, and so here are my references. And thank you for listening.